of Pharmacy, University of Jember, began with the establishment of the pharmacy study program based on the Directorate General of Higher Education, Ministry of National Education Number Decree 937/D/T/2011-2003 regarding the pharmacy study program operational permit. So May 7 is designated as the birthday of the pharmacy study program, University of Jember. Based on the approval letter of the Director General of Higher Education number 3910/D/T/2008 on 7 November 2008 and Director's Decree number 11057/H25/PS.8/2008 on 5th December 2008. Pharmacy study program was changed to become Faculty of Pharmacy, University of Jember.
The archipelago country of Indonesia is rich in natural resources, including the mega biodiversity. Bioprospecting for future prosperity should be conducted in accordance with environment conservation for mutual benefit. University of Jember is located in Tapal Kuta region, surrounded by the Regency of Lumajang, Probolinggo, Situbondo, Bondowoso, Banyuwangi, where most of the community occupation are related to farm and fishery. This geographical asset has led the University of Jember to have commitment as an excellent science, technology, and art, with elaborative aspect to the environment, business, and industrial agriculture. The vision of Faculty of Pharmacy is to strengthen the university commitment in the development and implementation of pharmaceutical sciences and technology related to environment, business, and agro-pharmacy. This excellency is driven through application of pharmaceutical sciences and technology in development of natural products and pharmacy services. Insightful to the environmental and business is reflected through environmental sustainability, competitive products which gain high economic value. And agro-pharmacy is based on the development of natural products based on pharmaceutical added value and development of pharmacy services for agricultural and coastal communities.
Faculty of Pharmacy, University of Jember currently has two study programs, namely the Bachelor of Pharmacy Study Program and the Pharmacist Profession Study Program. Currently, the Bachelor of Pharmacy Study Program has a rating accreditation based on the decision of LAMPTKS number 0090/AKR/SAR/2/2020. Meanwhile, the Pharmacist Profession Study Program is operated based on Minister of Education and Culture Decree number 37/E/O/2014 about Pharmacist Profession Study Program operation permits at University of Jember. Currently, the Professional Pharmacy Study Program has a B rating accreditation based on the LAMPTKS Decree number 0313/LAMPTKS/AKR/Pro/5/2017. Faculty of Pharmacy, University of Jember has several divisions and laboratories. First, Biology of Pharmacy Division consists of Pharmacognosy Laboratory, Phytochemical Laboratory, and Microbiology and Biotechnology of Pharmacy Laboratory. Second, Pharmaceutical Division consists of Technology of Solid Product Laboratory, Technology of Liquid and Semi-Solid Product Laboratory, Biopharmaceutical Laboratory, and Technology of Sterile Product Laboratory. Third, Clinical and Community Pharmacy Division consists of Pharmacology Laboratory, Clinical Pharmacy Laboratory, Community Pharmacy Laboratory, and Experimental Animal Laboratory. And the last division is Pharmaceutical Chemistry Division, which consists of Pharmaceutical Analysis Chemistry Laboratory, Medical Chemistry Laboratory, and Chemobiosensor Laboratory. In addition, Faculty of Pharmacy, University of Jember, also has a Medicinal Plants Education Center or WETO as a display media for Indonesian medicinal plants. In the future, WETO will be developed into an educational tourist spot about various kinds of medicinal plants in Indonesia. For supporting the education process, Faculty of Pharmacy has been equipped with various adequate facilities such as lecture halls, laboratories, educational pharmacy, reading rooms, computer-based test or CBT room, Wi-Fi hotspots, prayer rooms, parking lots, student organization and student activity unit building, and also a sports field. To support the research activity, Faculty of Pharmacy has formed several research groups or CRIs. Among others are Drug Utilization and Discovery Research Group, Chemo and Biosensor Group, Preclinical Pharmacology, Medicinal Chemistry, Drug Utilization Research Group, Drug Delivery, Bioactive Natural Product Development, Chemometrics, Biomaterials and Bioproducts, Active Pharmaceutical Ingredients, Modification and Cosmetic Products. The entire research activity supports the achievement of the faculty's vision, which is to become an excellent faculty in the agropharmacy fields. Faculty of Pharmacy also has several student activity units and student organizations to support the self-development
of the pharmacy student outside of lectures activity, such as Charisma Student Activity Unit,
Lingkar Pharmacy Student Press Organization, Philadelphia Christian and Catholic Student Activity Unit, Ashwa Islamic Student Activity Unit, Pasadena Student Sports Activity Unit, SMC Student Art Activity Unit, and Recording Nature Lover Student Unit. For the student organization, Faculty of Pharmacy has two student organizations, namely the Student Executive Board, or BEMF, Faculty of Pharmacy, and the Student Representative Body, or BPMF, Faculty of Pharmacy. Several achievements have been achieved by the students of Faculty of Pharmacy, University of Jember, such as winning a gold medal in Timnas 28 at Halu Oleo University Kendari, leading the Indonesia Pharmacy Student Scientific Week event, participating in national and international conferences, and many more. Not only students, but the faculty's lecturers also have very proud achievements, such as Professor Dr. Andes Bambang Kuswandi, MSc PhD, who's chosen as one of the world's top 2% scientists by Stanford University. The Pharmacist Profession Study Program educates students to become professional pharmacists who are ready to enter the community and give a pharmaceutical service in pharmacies, hospitals, industry, and government organizations. In addition to the lecture activities by internal lecturers and guest lecturers, the Pharmacist Profession Study Program also carries out several activities such as Professional Pharmacist Work Practice or PKPA, Computer-Based Test Inpatient Pharmacist Competency Vital Exam or CBT UKAI Tryout, CBT UKAI Exam Objective Structured Clinical Examination or OSCE Exam, Community Service, and Pharmacist of Thinking. Faculty of Pharmacy, University of Jember has collaborated with various institutions such as School of Healthcare Professions, University of San Carlos, Philippines, University Science Islam Malaysia or USIM, Leibniz Institute for Plant Biochemistry or IPB Germany, and the University of Wollongong, Australia. For the PKPA program, Faculty of Pharmacy collaborates with Tawangmangu Center for Research and Development of Medicinal Plants and Traditional Medicines, B2P2TOOT, Consumer Complaint Service Unit, National Agency of Drug and Food Control, Lokapom Jember, Pharmacy Installation District of Jember Health Department, hospitals, health centers, industries, and pharmacies. Faculty of Pharmacy has thousands of graduates or alumni spread across various fields of work and education and become members of Alumni Association, ICA Pharmacy, University of Jember. Several activities have been carried out by ICA Pharmacy, University of Jember, namely social services for the COVID-19 pandemic at Jember Regency Hospitals and Health Centers, the donation for victims of the eruption of Mount Semeru, and Munas ICA Pharmacy, University of Jember.
Ladies and gentlemen, welcome back to the last plenary session. Just like the past plenary session, this session will also consist of two presentations from the Honorable Sri Asutik Master of Science PhD candidate Technische Universität Dresden, Germany, and the Honorable Dr. Apotheker Muhammad Amrun Hidayah from the Faculty of Pharmacy University of Jember, Indonesia.
There will be a 30 minutes discussion that you can participate in after the last presentation. Alright, without further ado, let us start our last plenary session. With honor, we would like to invite Apotheker Afifa McLaurin, Master of Science, as the moderator for today's plenary session. Greetings, ma'am. How are you today?
Good. Can you hear my voice?
Yes, ma'am, loud and clear. Thank you. Before we begin, please let me briefly introduce the curriculum vitae of Apotheker Afifa McLaurin, Master of Science. Apotheker Afifa McLaurin, Master of Science, is from the Department of Clinical and Community Pharmacy, Faculty of Pharmacy, University of Jember. She studied in the Department Health Science, Unit Pharmacoepidemiology, Pharmacoeconomics, and Therapeutic Outcomes in University of Groningen from 2017 until now. Her areas of expertise are pharmacoepidemiology and pharmacoeconomics. Apotheker Afifa McLaurin, Master of Science, has published some research papers, and the last one is entitled Cost-Effectiveness Analysis of BCG Vaccination Against Tuberculosis in Indonesia, a model-based study in 2020. And that is a little bit about our moderator for today's last plenary session. Without further ado, to Apotheker Afifa McLaurin, Master of Science, the time is yours.
Okay, thank you very much. Good afternoon, ladies and gentlemen. So this is the last plenary section of this great international conference in medicinal plants. So let's start this plenary section. So the first narasumber here is, let me share with you the curriculum vitae of her. Okay. Can you see my screen now? Okay, so our first speaker now is Sri Astutik, Master of Science. So she is from the Cibodas Botanic Garden, the Indonesian Institute of Science, and she is now doing the PhD student in the Faculty of Environmental Sciences, Institute of International Forests in the Technische Universiteit of Dresden. And her areas of expertise is in the conservation biology, biomanagement, and the climate change. She already published many articles, and the latest one is in Asian medicinal plants production and utilization potentials as a review in Sustainability Journal, and also many journals in the last before years. And she also has awarded-based scholarship. There are two recent scholarships from the Reset Pro and also the GFF scholarship from the TU in Dresden. So let's get started with her presentation today. So she will present about the in the life, especially in the rural area, the conservation part. So this is very interesting. I think very interesting topics in our medicinal plants topics. So time is yours, Mrs. Sri Astutik, please.
Okay, thank you very much, Miss Afifa. Yeah, nice to meet you here.
Nice
and
to meet you.
Good morning, good afternoon, and I guess also good evening for all of you who are living around the world here. And thank you very much that I also can attend this international
Recording in progress.
Oh, wait a minute, please. I think I need to click something. Yes, international conference, medicinal plants, on medicinal plants. So it's a big pleasure to be here because finally I can present something or share my knowledge, especially about medicinal plants with my alumni here. I was also studying, especially in the Faculty of Agriculture. At the time, when I come again to or visit again in University of Jember, everything is changing. Ultimately, it's really different with some years back. So, yeah, it's really interesting also, and then I really love it, and I hope that I can go there again after I
Hopefully.
finish my study. Yeah, of course.
Yeah.
And thank you very much for all of the participants, especially that already here, and really happy to share what I have done in the last year, especially. And I mean, I have been working for another paper in this year, and I hope that I can finish my study next year. And it's been a pleasure that I can be here as a PhD student, still in the PhD student. Okay, then I can start, yeah, my presentation now.
Yes, you
So
can.
I'll try to go directly here, and I hope that it can
Yes, we can see very
it can
clearly.
be shown. Okay, so we can try. I'll try and then ask for your confirmation. Is that? But I think it's something. Wait a minute, please. Now, can Miss Afifa especially, can you see
Yes,
the
that's very clear.
presentation? Okay, thank you very much. So, well, everyone here, I would like to share, especially about the topic reconciliation of medicinal plants, production systems, and forest conservation in Java, Indonesia. That's why I tried to provide like two words here. Either it is options or challenge for us, because the idea behind of this presentation is about conservation. I would say vis-a-vis, yeah, not versus, but vis-a-vis, because if we say like something in versus means that both of them like contradictory. But what I mean here is because we have in one side about the conservation, but on the other side we also have development. So the idea behind of this presentation is about both of two words, conservation vis-a-vis development. So let's start from the first slide. I apologize. I think something happened here. I couldn't. Yeah. Okay. And today I would like to share with you, especially some sections, start from introduction until a call to action. So it's quite huge topic, but I hope that I can deliver it in a good way to you and in the concise content also. Then we can start here. I use three keywords here. Start from the conservation and sustainable use and also production systems. Why I use these three? Because we try to work in term of conservation, and then we use medicinal plants sustainably in term of preserving all of the species and ecosystem. I mean, in the level of genetic and then species and also ecosystem of medicinal plants. Then we can also plan it, or even we can also use in the daily life. So that's why we also work with this kind of term. Perhaps some of you or even all of you already know about the production system here. So this is what I have been learning during my PhD journey in Germany. So I think I don't need to read all of them, but at least you know the highlights. What is the conservation, sustainable use, and production system? So it means, in my point of view, when we talk about conservation, it doesn't mean this is something rigid, but we need to look at it in the flexible way. That's why then we shouldn't just only stick on the point of view that if we conserve something, so we couldn't do anything. To me, this is my personal opinion. This is we need to see. We need to see that the conservation is not something that we couldn't do anything. That's what I mean. And then we can go to the next one. Yeah, I try to see. I mean, like to find some like, let's say, like to picture here. And as you see here, that how the people try to imagine conservation and development, looks like that both of them in a contradictory level, right? And then, so in this case, I would like to bring all of you in the context of our era right now, because we know about what the people or around the world, or the global commitment to launch sustainable development goals. I hope that you're already familiar with this concept. So it means that we need to bring together on our table how the people can do conservation, while on the other hand, we are still doing a development, or even try to focus on the development. So how could we make it in balance? Then the next one, especially for medicinal plants, we would like to see in the context of development agenda. Then now we can see like from some point of view, start from the inventory and standard forest inventories, and then we also need to coherent research, which is focusing on the ecology and sustainable management. Both of them should be done in the balance situation, or in the balance circumstances. And then we also see that we can also create like medicinal plants division, especially at the rural area. And the next one is about the development plans. As you already know, that I explained that this is like trade-off decision. How we can bring the conservation of medicinal plants together with the production system of medicinal plants, production system management of medicinal plants. So then the next one, we can see here, we need to include, or even to put it together in the sectoral policies. Medicinal plants, of course, is an important resource, especially Indonesia. We have, I guess, I'm afraid if I'm wrong, but as far as I know, that we are still in the top level about the biodiversity, especially our position around the world. We are still in the top level, at least in the top 10 of biodiversity around the world. And then especially the next one is about the security of tenure, land, and access on the land use system, I mean. And then case by case, so it means that we need to see what kind of medicinal plants management that we can manage in each area. So it means that, for example, in Java, the medicinal plants or Javanese medicinal plants could be different with the management in Sumatra, or even in another area in another continent, like, for example, in South Asia, I mean, like India, Nepal, or even in Mexico, America Latin. So every place they have different way to manage how to conserve and to develop or to manage medicinal plants. And then we can see, I guess all of you know that medicinal plants can be used for two things, especially for healthcare purpose and for our daily life. What I mean here, we can also see that from the here, I'm sorry, I try to put it like this. Yes, we can see that, for example, you see that this is curcuma, right? Curcuma, or even for ritual also. So it means that medicinal plants can be used in almost all of our daily life. Start from the like condiment. Perhaps you know, some of you already heard about satay, maybe. Someone or people who are living outside there. So in Indonesia, we have quite famous cuisine. We call it satay here in the first picture. So we use many like spices. Yeah. Sometimes we also put condiment as a medicinal plants also. And then even in the ritual purpose, we also use some medicinal plants. Then we can go to the next level. yeah, as we already know that for healthcare needs, the traditional medicine, there are some system there. As you already know, also we have Ayurveda, traditional medicine. In Indonesia, we have Jamu, and like Sawarikpa, it is from Bhutan. Yeah, correct me please if I'm wrong, but as far as I remember, it is the system, traditional medicine system in Bhutan. And this is a special case that for livelihood security, as you can see that based on some studies or even many studies in Asia, that medicinal plants can generate cash income for the people, especially the people who are residing around of the forest, or even they who are living around of or even in the rural area. The contribution on the household income more or less is about 3 until 44%. Is quite high, almost half, a half of their income in the household level. Then in this case, I would like to reinforce the concept, especially medicinal plants. How could we see the medicinal plants, especially in the era of sustainable development goals? So both of them, conservation and sustainable use, create ideally as a closed loops. So it means that both of them should be in the same loop. Both of them should be in line, not a contradictive, not something contradictive, but this is both of them should be bring together. So we need to harmonize people, planet, prosperity, partnership, and peace. This is like the so to say, like the quite a big vision of the global life right now, that we need to bring medicinal plants in the context of a sustainable development goal. Then the conservation here, especially in the context of sustainable development goals, we can see from both of sides, either production system and methods. So I would like to underline, just to let you know, that production system, which I also have done, it's about wild gathering and cultivation, based system. It means that people can also work in the forest area for collecting medicinal plants. Just invest by investing their effort and time, labor. So, but they don't need to plant it. So it means that this is like the people just harvest it from the wild area, especially in the natural forest, and then the cultivation. We need quite, so to say, we need quite a lot of investment, because we need to invest labor and then cost, another cost. I mean, many other things, transport, and I would provide some data based on my case study to you for afterwards, and then especially about the financial performance that I already calculate. So then we can understand. So we can compare the effectivity. What I mean here, like let's say the performance, comparative analysis between wild gathering and cultivation-based system. And then the methods, as you already know, either in the in situ area or ex situ. So I have been working for a botanic garden. So it means this is the example how the people conserve medicinal plants in the section of ex situ area. While here, especially you who are living in Jember, you already know about Merubetiri National Park, for example. So this is the example of our in situ conservation. Then we move to the next level. So as I already did last year, this is the result of the review paper. I already published it, and then as you already know, based on this data, means that nowadays in Asia, especially the trend of the research, especially in terms of medicinal plants, people try to a bit move move forward or shift the production system to cultivation. Yeah, even though we also still have a lot of people who are working with forest gathering, but nowadays the trend is becoming more. So the people try to cultivate more about medicinal plants, whatever the species, especially the species you usually can find in the market. So many other countries, and especially in the area I already mentioned here, Southeast Asia, and China. So the literature I found already said that there are some cultivation production system which people already try to focus on. So this is a good sign that finally people realize that we need to cultivate, not only to just grab it or even like directly collect it from the forest, but we need to cultivate it. It's a good sign anyway. Then this is the status in general about Asian medicinal plants. I just to let you know why, because when we would like to break it down into the case study, in for example, in my case is about Java, Indonesia, means we need to know what is the state of the art of Asian medicinal plants nowadays. So for the 5 lists here, as you can see that the especially Asian medicinal plants now, we have 5, we have 5 main conclusion from continuous shift from wild gathering to cultivation, and then the potential as traditional healing, and then livelihood support, also modern healthcare. And then we also see that fragmented knowledge is still happening there, either from multi-scale institutional, formal, or even informal here. And this is interesting, especially for the 4th one, very few studies. Yeah, this is a challenge. The challenge of medicinal plants commercialization. I hope that I can compile or even find any kind of data which is explaining in detail about what is happening basically about the commercialization of medicinal plants around the world. But it's quite difficult, So it means that it's still. I mean, we don't know from where we take this kind of species, and then suddenly we can find in the market this happening. And then the last one is about law enforcement, and then benefit and knowledge sharing, and also research and development. The next one, So no, we are coming to the case study directly. So I use the island of Java. As we are already here also in the part of East Java, especially in Jember. So what I mean by PS here is production system. So I try to use 4 villages here. The first and the second one here, I'd like to highlight here. It is located in the Meru National Park, as you can see here. I hope that all of you here already familiar with this place, Sanin Rejo and Andong Rejo. As I hope some of you also already know that quite famous villages here in Jember, which is focusing on the medicinal plants management. And then the second one, the third one is outgrower production system. It means that the people try to cultivate medicinal plants, and then they try to cooperate with industrial level. So that's why I choose Java as a case study in my research. As you see also, there is a limited comparative studies of production system linked to local livelihood especially. So, and we can also see, this is what I mean. In the very first time I mentioned, this is a calculation that we can compare between three system here, 3 production system. then I just point out directly, especially in the level of net benefit, net benefit per production area. As you can see here, this is in the household level. This is in the household level, means that forest gathering can contribute. Sorry, I would like to highlight net benefit. Yes, point H. I would like you to just focus first in the H. In the point of H. We can see the net benefit here, the highest one. As you can see here, we can obtain or the people can receive quite high. Let's say 597.89 US dollar per year. So compare with forest farming and farm-based outdoor production. So it means that it is undeniable that forest gathering can contribute more than cultivation system, right? So that's why it is our big homework that we need to solve. Because if we provide, if we allow the system go on, means that we need to make sure that the species which are collected by the people or by gatherer can be how can I say it, can be sustainable in the long run. But we never know about it, right? So we never guarantee that the species is not gone there. The species is still there, so we couldn't guarantee about it. So this is like the alarm or red alarm for all of us. How to make it sustainably for the long term. So then, but as you already also see here, when we come to the net benefit per production area, means that we count it in hectare. So we can see that cultivation can provide more than gathering system or wild gathering system. So because in this case, if we work with forest gathering, means that we work with a lot of people, a lot of gatherer, which are visiting or coming to the forest, and they collect it together and work together in a group. So it means that the people can use, like, let's say this is like pie, like a pie. Then the people try to eat or even try to use all of the area in pie. So means that that's why it is divided by the polygon or the area, the work area of each of the people or gatherers. So that's why then they obtain like the less number or benefit compared with the other system or cultivation system. Okay, then we can go to the next one. This is what I can provide it to you. When I asked the people, especially in the tree of the system, we can see almost all of them say that medicinal plants can contribute to their livelihood moderately. So it means if or in case we can manage it in the ideal situation or ideal level, it could be shifted into the next, to the left, like high or especially or even extremely high. But it is easy to say, not easy to do. So in this case, I just would like to highlight that basically people are still depending. Especially in my case, people are still depending their income from medicinal plants. So now I come back to the what is that, like the huge issue about the SDG. I tried to open this presentation with the SDG in general, and then I tried to provide a case study, and then now we are coming back to the huge issue again about what is the point of SDG related to medicinal plants conservation and development. So we need to bridge conservation and development together. As you can see here, so we can work in 1, 2, 3, 4, 5, 6. Yeah, in the six point of SDG, and how could medicinal plants can be can be a good resource or even like effective resource to handle, especially or to support in the achievement of all of this SDG point that I already mentioned here. Start from poverty issues, as I already mentioned to you, that finally people are depending moderately. I mean, depending on medicinal plants moderately. So it is a good sign that medicinal plants really important. Medicinal plants are really important for tackling the poverty issues and good health and well-being. Also, that we also have many traditional medicine system around the world. So it means this is a good resource also to work with. And then responsible production and consumption, and especially climate action. We also can use medicinal plants for this kind of function, climate action, because we have many types, many types of habitus of medicinal plants. Start from the tree and then shrub, or even we have herbs. So it means that we can use it. We can use it for tackling climate action. Life on land. This is we talk about it, especially in the level of landscape. So it means that we need to coordinate with another management. So maybe I need to mention here, because in Jember we already have Blambangan. What is that? Blambangan Biosphere Reserve. Yes. So this is really related with the issue, the SDG 15. It is about life on land, so the people can manage medicinal plants based on the level of biosphere reserve, not only in the household level, not only in the rural area, but we can manage it in the landscape level. And then partnership for the goal. So everyone should be involved in this kind of activity. Yes, this is the legal platform that we can refer about medicinal plants management here. And I guess that all of you already know, because some of them, I'm sorry, I hope that I can write. Yes, several of them is like national regulation, but many of them are global. Many of them are global regulation. Let's say like Convention on Biological Diversity, and then national strategy for sustainable development, and we can put like goals. On climate change. So it could be in the next level or the next group, the people also can work based on based on the climate change issue. Okay, then the next one. Yeah, this is a potential challenge we have. So it's better that if I don't mention all of them, but the basic thing here that we need to combine four of them. Start from environmental issues, economic issues, socio-cultural, and also institutional. So as you can see here, so we need policy coherence, how to bring it together. Because if we try to focus only on the conservation, means that we don't allow people to produce it either from well gathering or in cultivation. Okay, but from well gathering, this is the quite sensitive issue, because nowadays people are still depending a lot in the natural forest. I hope that you can You can understand what I mean, because this is the natural. I mean, this is the real fact we already see today, that the people come to the natural forest and then they just gather or collect all of the space. The commercial species, and then they sell it. But it has been done during, like, let's say, for many years, or even 100 years. And especially here in Jember, as far as I know, that they have done it even before the Republic of Indonesia is established. So it means it's like the let's say, like traditional. How could we say, like traditional heritage, that the people try to maintain this what is that, like this activity, and hand it down from generation to generation. This is a big homework for us, how to manage it in a sustainable way. So for the next one, yes, this is another point of future challenge. So I would underline, especially here in the economic issue, and for the people who are working with medicinal plants already know that we should concern, especially about the upscaling of the business, the business of medicinal plants, not only like in the small business, but we can also upscale into the higher level. Like, even though SMA of course contribute a lot to our national economic, but what I mean here, how to upgrade it in the like in the ideal level. We need more. We need more resources, of course, and then product certification, especially. This is really important point. So then we can guarantee and we can assure to the people that what we already sell to the market, especially medicinal plants, is also safe to consume. Is also in the good condition. So I just would like to share a bit with you, because I have been living here in Germany for, let's say, like quite some years. So I can see that, for example, like when they try to sell the herbs, like herbs product, they are really concerning, especially about this one, product certification. And the people never ever sell something without any certification. So that's why then the people feel safe when whatever and whenever they buy the product. I mean, like herbal products, they feel like safe because it's already in the good quality, or even So people can just assure that, okay, then I can just drink it or even just eat it safely. Okay, then
Yes,
I think
remind you. Okay, thank you. So the time only maybe
1 minute or 3. Okay, almost finished, Miss Afifah.
Okay.
Yes. Yeah, this is sustainable management of medicinal plants. I already published it. So this is my opinion, or even my suggestion, or even what I mean here, because we have done it in the team. So we suggest this kind of scheme, how to manage medicinal plants sustainably. So we need to bring all of them together. From commercialization, conservation, and institutional. So we need to bring it together on the table. So we don't need to contradict between 3 of them. And then we come to the next level. Yeah, quite a lot, call to action. So domestication, and we need to conserve our forest also. And the next one is about, especially, of course, about the knowledge. Knowledge of medicinal plants. We need to what is that? Like, we need to maintain this kind of knowledge. So young generation, I hope, especially the student. I really appreciate the student who already learn about pharmacy, especially pharmacy and some related field. Is really good. And then we also need it in the long run. And about unfair trade, pros and cons. We need to concern also about it in the international, global, and national trade. And yes, this is the very last quote that I really like to share with you. Perhaps you know Professor Emil Salim. He ever mentioned this one: a wise eco-development is urgent for a better human dignity and quality of life. Yeah, so means that both of them we can marry between eco-development and conservation and development. Both of them are in line basically. Okay, that's what I can deliver to you all, and thank you very much.
Okay. Thank you very much, Mrs. Sri Astuti, for the great presentation. Let me wrap up, as our world is only one and it can't be enlarged. So the conservation and the development is like two important things in sustainability, that it can rise many challenges, but in other side, it also has some potential. So maybe we can discuss in the discussion part, but let me introduce also our second speaker here. Let me share with all of you his curriculum vitae. Okay. So can you see my slide well? I'd like to introduce you to our next second speaker is Dr. Apotheker Muhammad Amrun Hidayat. So he is from the Pharmaceutical Biology Department of our Faculty of Pharmacy, University of Jember. And his last education is in 2022 in University of Erlangga as doctoral of pharmaceutical science. And his areas of expertise is pharmacognosy and also optical, chemical, and biosensor. And his latest publication already published in the 2022 about the paper-based sensor for some biomarkers. And he also awarded some numerous research grant as a reviewer also. And so without further ado, we will present about the very interesting topic also about the application of paper-based sensor on food, beverage, and medicinal plants extract determination. So this is also interesting in our medicinal plants knowledge. So let Mr. Amrun, can you hear me? So maybe
Yes.
you can unmute. Okay.
Yes.
So not waiting for too long, just begin the presentation and time is yours.
Okay. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. First of all, I would like to appreciate the Committee of International Conference of Medicinal Plants that give me the opportunity to share my research for so long. And without waiting again, please let me share my PowerPoint. Is everybody can see my PowerPoint?
Yes, very clear. Yes.
Okay. So today I will talk about application of paper-based sensors on food, beverage, and medicinal plant extract determination. First of all, we have to know what is a sensor. Sensor is device that detect events or change in quantities and provide a corresponding output. We know that a mercury thermometer is an example of a physical sensor. It converts the measured temperature into expansion or contraction of a liquid, which can be visualized on the calibrated glass tube. Another example is thermocouple, which convert temperature to an output voltage that we can see in the display. So there are two types of a sensor. First of all, is a physical sensor. Physical sensor is a device that provides information about physical property. And for example, is pressure sensor, temperature sensor, etc. And secondly, is chemical sensor. It is a device that transforms chemical information, ranging from the concentration of specific sample component to total composition analysis into analytical signal. Example of the chemical sensors is carbon dioxide and pH and ion meters. So we go more deeply about chemical sensors. It is defined as an analytical device incorporating a chemical material or region that can recognize or react with chemical analytes, whether in the solution or even in the atmosphere, with a physical transducer. So a transducer is maybe look like a detector in a chemical instrumentation. So in concise, chemical sensor consists of a region plus transducer. So what about the biosensor? Principally, biosensor is analog with chemical sensor. The difference is it is used as biological material as a biological or biorecognition. So it can recognize biological or chemical analytes in any type of sample. So the example of a biomaterial or biorecognition is enzyme. Mostly the biosensor is developed by using enzyme and also antibody. So, and DNA, even whole microorganism such as E. coli can be used as a sensor or biosensor material. So in concise, biosensor is made by a bioreceptor plus a transducer. So today I would like to share my expertise and my experience for developing paper-based sensor. So in historical view, a litmus paper is basically a paper-based sensor. It measures pH. So if we have acidic solution, it converts blue litmus, red litmus into blue color litmus, and so on, in opposite. And then secondly, in the medicinal plant fields, we already know that chromatography paper. So basically a paper chromatography, which we can use as a spot test for identification of many compounds and phytochemicals of medicinal plant is basically a paper-based sensor, which is a simple paper-based sensor. So why we use a paper? First of all, paper is a good medium of spot test because it is white, which provide a strong contrast with color test. Of course, white background is more desirable than a black background. Secondly, it has a high surface to volume ratio, which facilitates accelerated evaporation of solution. In this case, whether a region or bioregion results in a concentrate analytes or a region or bioregion on the sensing zone. Thirdly, it is extremely cheap and easy to handle. So everybody know papers, everybody work with papers. So everybody can make a paper-based sensor, actually. And it can be designed as a lateral flow, just like a pregnancy test or flow analysis that run by capillary action without external pumping, such as, for example, high pressure liquid chromatography and so on. And lastly, it is disposable and more eco-friendly than plastic-based cuvette or microwave plate. So here is an example of glucose sensor and protein sensor. It is to analyze glucose and protein in urine sample simultaneously. So if we ask what is the application of the paper-based sensor, so if we look at Indonesian herbal pharmacopoeia, there are several tests for phytochemicals of medicinal plant extract. Number one is matter compound detection. Let's say, for example, like quercetin or maybe sinencetin. Secondly is total group of compounds. Let's say as a total polyphenol content and also total flavonoid content. And number three is a TLC profile of a single extract or raw material, or as a simplicity. So can paper-based chemical sensors or biosensor apply to this chemical test according to Pharmacopoeia Herbal Indonesia? So in my research, I always use paper-based chemical sensors. I developed what is called a paper microzone plate as a select support. So it is firstly introduced by Professor Whiteside group. It is used as an alternative for plastic-based 96 microwave plate. So basically, it is a filter paper, which is screen printing by using rubber ink to make a circular pattern at both sides of the paper. So basically, for sensor development, the region or bioregion, which in case in the biosensor, are immobilized on each microzone as a sensing zone. And then we have a standard or sample solution is applied, is added to the dried region of bioregion. And then the color chain of the sensor is captured by a flatbed scanner here and further quantified by image processing program. For example, imagery, which is known this technique as a scannometric method. You can see what is scannometric method in my publication. So the first sensor I would like to share with you is single marker detection, or in this case, quercetin sensor. It is inspired by the work of Askal et al. In 1997. So if we have a flavonol, it can be analyzed spectrophotometrically using N-bromosuccinimide. Fortunately, this region is reacted selectively with quercetin, forming a deep violet color, which can be analyzed at 510 nanometers. Whether the other flavonols, such as camphorol, or other like luteolin, is not forming purple color with NBS. So what happened here, quercetin is reacted with NBS to form its bromo derivative, which has a purple color. So we adopted this chemical reaction into the paper-based form. So in paper microsome tape, I immobilize this NBS region, and we mix the standard quercetin or sample extract with 1% phenol to stabilize the pink color, which is arise after the sensor is reacted with a standard or sample solution. This purplish red color can be quantified as the analytical signal. Here, this is a blank sensor, and this is a quercetin in a concentration ranging from 1 to 10 millimolar. So, secondly, if we look at WHO, there are tests that is recommended by WHO, such as heavy metal contamination, and what I also developed a formaldehyde sensor, basically as a paper chip for formaldehyde detection. It is actually a biosensor which use enzyme, alcohol oxidase, which can oxidize formaldehyde, and the oxidized formaldehyde further reacted with tetramethylbenzidine to form a blue-yellow color, which can visually detect it, or we can quantify it using imaging program. So after we conduct research, this chip can be applied for formaldehyde detection in noodle and meat samples. So I'm sorry, this is a wrong place maybe. The quercetin sensor used for quercetin determination in guava leaf extract sample. We know that guava leaf extract is a starting material for jamu or herbal, standardized herbal medicine, and also phytopharmaca. So measuring quercetin is very important. And our research show that the developed sensor can result, the result of development sensor for quercetin determination is not quite different with the standard TLC densitometric method. Okay, and then the second application of paper-based sensor is for total polyphenol content. In this case, I could the Archeoli et al. 2013 for the developing a biosensor for total polyphenol content. It is used as tyrosinase enzyme and chromogen MBTH. This enzyme can oxidize the substrate, monophenol or orthodiphenol into its quinone. And the quinone compound can further react it with MBTH to form pink colored agar. So in their research, the method is developed for PPC determination of line sample, expressed as L-DOPA equivalent. Another example of tyrosinase and MBTH-based biosensor is a biosensor for determining many polyphenols such as 4-chlorophenol, catechol, M-cresol, and paracresol in the water samples. It is basically the same mechanism and use this very same bioregion and chromogenic region. Here, dopamine as used as L-DOPA is used for the biosensor characterization. So our publication use very same mechanism of biochemical reaction. We just use enzyme, but I'm sorry, we use catechin as a standard for validated sensor. Sorry, should be catechin. So here, if we have a sample containing catechin or polyphenol, it can change the colorless biosensor into red color biosensors, and the color change can be recorded using a scanner, ordinary flatbed scanner, and the color intensity can be quantified using an imaging program. And this developed method is applied for green tea beverage. So our research is different with the previous biosensor research, that we use a catechin as a standard, and to express the total polyphenol content, we know that catechin is a basic structure of green tea polyphenol. So it is more related in the core sample than the previous biosensor. We know that L-DOPA is not related with the wine sample in the previous research. Okay, the next application, we use a chemical sensor for total polyphenol content. Basically, it is used sodium periodate to generate catecholamine. So sodium periodate is an oxidator, which is used as a oxidator to replace tyrosinase or to study kinetic of a tyrosinase enzyme for many polyphenols such as catechin, chlorogenic acid, etc., etc. And it is more stable than tyrosinase, and the reaction is speedier than the tyrosinase. So we developed the sensor in the form of a street test, and the developed method is applied for the TPC determination of green tea beverage. And TPC is expressed as catechin equivalent in ppm. Here, the result show that our developed method, the result is quite similar with that spectrophotometric method. So the similar platform, the similar chemical mechanism is also applied for total polyphenol content in the coffee samples, in coffee beverage samples. But the difference is we use chlorogenic acid as a standard. We know that CGA or chlorogenic acid is the main polyphenol found in the coffee. So we use it as a standard, and we calculated the TPC in milligram per liter chlorogenic acid equivalent. So basically, the biomechanism or the biosensing mechanism is the same. CGA is oxidized by sodium periodate to form CGA quinone, and the CGA quinone can react it with MBTH to form the CGA quinone MBTH complex, which has pink color at the sensor surface. So our result show that the developed biosensor can be applied to many coffee samples such as Arabica, Robusta, Green Blend, Grown, and Mogus or Luwak coffee. Okay. The next application of pepper-based sensor is antioxidant capacity. We differentiate antioxidant activity with antioxidant capacity. If we tested the single compound, we call it antioxidant activity. But if we test the multi-compounds such as extract or fraction, we call it antioxidant capacity, or in other literature, often called total antioxidant capacity. Here, I show you the example of the micro pepper analytical device. It is fabricated by lamination method using ordinary plastic in plastic film, the plain plastic film, which is we made some hole and filter paper inserted between two plastic, and it is laminated, and UPPH is immobilized on the filter paper and to use as a sensor. Here, the sensor is show a linear response toward gallic acid, and the sensor applied to green tea samples. So we also made PPH-based sensor, but instead using laminated film, we use the similar pepper microzone plate as our previous publication. So basically, it is made simply by immobilizing PPH into the pepper microzone plate, and then the standard or sample solution is simply added to the sensing zone, and the total antioxidant capacity is determined or calculated as millimolar gallic acid equivalent. So this antioxidant sensor also applied to many plant medicinal extract such as West Indian elm leaf, red guava leaf, white guava leaf, stone breaker leaf, kutipling or green tea leaf, and this is a Jati Belanda. And then the next application of antioxidant sensor is it is based by cupric region. We use the same pepper microzone plate, but we change the region. Here we use the cupric region, which consists of neocuproin, ammonium acetate, and cuprochloride. We immobilize the cupric region into sensing zone. And after that, the standard odd sample solution is simply added to the sensing zone. And here we can see the blue copper reagent. If reacted with the antioxidant sample, it turns into yellow color. So the total antioxidant capacity calculated as millimolar routine equivalent. We also tested many medicinal plant extract such as turmeric rhizome or kunyit, guava leaf extract, whether red variant and white variant, and also black tea leaf and green tea leaf extract. And the results show that the developed method show no different observed between two methods statistically. And then maybe the last application of A paper-based sensor is a sensor array. Simply to say, sensor array is multiple sensor that contain different regions that used for simultaneous determination of many or different purpose. Here, at the Luktafon and Maka 2016, the total polyphenol and total flavonoid and also antioxidant capacity can be tested simultaneously by using paper-based microfluidic devices. Basically, it is choline Q and aluminum chloride and DBPH are individually immobilized onto the sensing zone here. And then after the standard or sample solution added to this sensing zone, the sample or standard solution is that the sensor is dipped onto the surface of the solution, and then the solution is moving by capillary force, and then reacted with the region, each of region. So simultaneous determination of TPC, TFC, and antioxidant capacity calculated as milligram carric acid equivalent, quercetin equivalent, and DHT equivalent. So here the calibration curve of total polyphenol, total flavonoid, and antioxidant capacity by using the sensor array. And then the next sensor array is a paper-based device with cross shape. It is fabricated by screen printed technology using polymer, which is a hydrophobic barrier. So the LBTS and cuprack and choline region were deposited on the pre-treatment and the detection zone here. Basically, LBTS and cuprack and choline each of all of them is consisted of two regions. For example, LBTS, here LBTS, they immobilize the LBTS and calcium and potassium persulfate here. And cuprack, it is the cupric chloride and ammonium acetate and neocuprine immobilized separately here, and the choline and sodium carbonate also immobilized separately. So this sensor work that if we put sample solution or standard solution here, the fluid or the liquid is moving through all the detection zone by using capillary force, which facilitated simultaneous detection of antioxidant capacity by two methods. It's actually ABTS and the cuprack as antioxidant capacity and total polyphenol content simultaneously. So here the sensor array is applied to determine TPC and antioxidant capacity of a tea sample. For example, oolong tea, black tea, and red wine sample. And the last one is my research is called color profiling using antioxidant sensor array. Basically, it is consisted. The sensor array consisted of the three different regions: the cuprack, the IBTS, and choline which is immobilized independently and multiplied here 3 times, and different sample, for example, guava leaf, red variant, or guava leaf, white variant, and turmeric rhizome extract is added here. So after several times, actually in second to several minutes, the sensor array change the color. Here, every extract has a different color profile. So this is the actual color of the sensor array. This is before the sensor array reacted with the sample, and this is the color of sensor after reacted with different samples. If we analyze this their color intensity, which is red, green, blue, and average red, green, and blue, we put it into analysis, multivariate analysis. We have what we call a color different map. Color different map is a substitution. If we measure the color intensity of the reacted sensor array, subtracted with the blank sensor array, we have the color different map. And the color different map is further analyzed using HCA, hierarchical cluster analysis, and also principal component analysis. The results show that every sample, each sample is clustered individually and clustered with their own type. So here, the GM sample or guava leaf red variant. This is the guava leaf white variant, and then this is a rhizome. This is a turmeric rhizome, and this is a guava leaf red variant, and this is a guava leaf white variant. And if we look at the principal component analysis, we know that all the sample are placed or located differently in the PCA score. So the conclusion of what we have done so far is, firstly, paper-based sensor has a long historical use in chemical analysis, while its practical use is continuously expanding in chemical and pharmaceutical field. Secondly, the paper chemical sensor, along with scannometric techniques, can be applied for determining a single marker, TPC, TFC, and total antioxidant capacity in food, beverage, and medicinal plant extract. The developed paper-based sensor with the scannometric technique is simple, easy to operate, and more economical than the standard method, which is suited for the lab with limited resource and for the on-site analysis or field-safe analysis. Here the reference we have published in some international journals. Yeah, and then thank you everybody. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for very interesting presentation, and also I think paper-based sensor is simple yet promising. I think, yeah, for especially in the accelerating the finding of more potential medicinal plants in maybe, for example, in the forest, because we can imagine that you only have to bring your paper. Instead, you have to bring your spectrochromatography. So you just bring your paper and you can go everywhere in the world, and then you can find potential plants for our medicine. So this is very interesting topic, and maybe we can discuss in the discussion part. For the participants, please raise your hand if you want to ask to our speaker today, for Ms. Astuti and also Mr. Amrun. Yeah, let me see. So still there is no one. As we can see in the chat room, it's already a discussion there. but maybe we can also repeat it again lively. Let me read the first question for Miss Astuti from Mrs. Ari Nurhayati. Yeah, I think it is a very good work of you. I'm very much agree with your idea. However, now we need more of the coherence between political and grassroots movement to increase health, conserving the traditional heritage. However, one problem should also think is the environmental sustainability, including to conserve the soil itself. Maybe Ms. Asuti can repeat again your comments and also your insight regarding this idea.
Okay, thank you, Miss Afifa, and thank you also to Miss Ari Nurhayati. Yeah? Nice to see you and your question also, and all of the participants, of course. Yeah, I think once again, thank you very much for your appreciation. And then, yeah, what you said is already right. That yeah, it's challenging. It's never ending work, you know. So because we need to work with all of the section, right? As I already mentioned, that we need to make it balance between institutional level and then economical area, and then environmental and social culture. So it's really easy to say, but it's not really easy to do. To execute it, because, for example, in some cases, based on my review paper, I already tried to see. Sometimes it doesn't. How can I say it? Like there is a gap. Yeah, there is a gap between, for example, like when you try to apply the policy in the local level or even the regional level, it doesn't link to. I mean, it doesn't. How can I say it? Like we couldn't see the line between the policy which is applied in the local level and national or even in the global level. So this is a huge challenge. So we need to make it in balance, or even try to fulfill the gap. But my suggestion in this case, especially we can start, especially in the regional level. It would be effective, because regional level can understand well about their area. For example, in our case here in East Java, so it means that the government in the East Java can coordinate or can encourage all of the stakeholders under the under the authority, and then each of the stakeholder can provide the let's say like their opinion or even their insight, or how they can manage their resource based on the local specific level. Yeah, let's say the government of East Java can coordinate with Murubetiri National Park, for example. So it means that we can focus on, and then of course with the government of Jember, for example. So it means that if all of the stakeholder can work together on the same table, on the same page, also how to see what is the problem basically. So then we can also find the solution. Yeah, even though it's not really easy, but at least we can do it. So that's what I can, and especially what is it? I hope that it can answer. I mean, it can be the answer. Thank you.
Okay. Maybe Mrs. Ari want to reply to the comments. Okay, let just going to Mr. Amrun, there is also a question from Fajrin. Uh, how has the application of this sensor, how has the application of this sensor been in practice until now? Is it applicable if you want to make it in large numbers, such as in industry scale? For example, maybe you can answer this question, Mr. Amrun.
Okay. Definitely, I already answered the question from to Ari. Yes, it is. Scanometric is basically is a colorimetric technique which measure the color intensity change after the sensor or biosensor is reacted with uh, the sample or the standard solution. So the question from the Upajrin, how has the application of the sensor has been practiced until now? Frankly to say, our developed sensor, the stability is very low. Let me show you. Let me show you example of perhaps I want to share screen. Is everybody can see here?
Yes, can see.
Yes. Okay. Here the example of a biosensor which is used as a total polyphenol content of green tea sample. Here we know that from the figure 5, the stability is held for only 9 days in freezer temperatures. Whether it is stored in a chiller temperature, it's only stand for 5 days. What about when it's stored in the room temperature? It is not lasting more than 1 day. So yes, I have to confess that the stability of the chemical, the pepper-based chemical sensor and biosensor is very limited. It has to be stored in the chiller or the freezer for the better performance. So in the industrial upscaling, I think I would like to say I don't dare to to scale up the sensor before we have to sure that their stability is very high. For example, as a pregnancy test, pregnancy test is a actually they immobilize the the antibody for the hormone human gonadotropin, which is stable for a couple months. But in our work, the stability is the major problem that we face here. So we cannot apply this the developed sensor. We have to make it recent apparatus, or we have to make it new, and we have to store maybe for the filtration analysis we have to store it in a dry ice box, or we have to make the sensor new. So we separate the region of bioregion in the bottles, and we made the sensor immediately. And yes, but in a practical point of view, it is can be used a field trip study, which is I think maybe it can help Dr. Sri Astuti in the field trip to detect on the screen what plant contain flavonoid, what plant contain polyphenolic, etc., etc. By using very cheap instrument. We have only use the ordinary office scanner or flatbed scanner. More recently, we also can use our smartphone and a free program, Android program, to measure color intensity. Almost every Android program which can measure color, we can use it as a interface to calculate any samples or any analytes indirectly. Thank you, Dr. Afifah.
Okay, thank you. Okay, it's also rise another challenge, I think, but maybe it can trigger future research for the next research, and hopefully we can have paper-based sensor that really stable, and then we can bring everywhere. Okay, there is still a question for Miss Astuti from Miss Puspitasari. I think you also already answer in the chat room, but because there is still no one raise their hand, maybe I just
Miss Afifah, I'm sorry, can I just interrupt a bit? I
Yeah.
think I see Miss Ari Nurhayati say something there. Could you see that, please?
Okay. Please, Miss
Still unmute maybe,
Can
or
you
something?
unmute yourself?
Yes, yes. Now I can. Yes. Okay.
Can hear you.
Thank you. I'm asked to Mrs. Astuti. I would like you also, but and then how to make like you work in Germany, is it now? Is it?
Study basically.
Yeah,
Miss
yeah.
Nurhayati, yes, study.
Yes. Yeah,
Yes.
yeah. What I think is the difference between Germany and here is what I mean is for how to conserve the soil itself in Indonesia,
Okay.
because sometimes the difficulty is the physical soil itself. Some kind of people assault the soil, but actually the soil is good. Is no heavy metal contamination in the soil, and then after that they because they need the money and then they sell the money. So it is difficult for the for the how to make a sustainable of the soil, also to conservation of the soil. This the problem in Indonesia also.
Okay.
Yeah.
Yeah. Yeah. So, can I just say something,
Yes.
Miss Nurhayati? Yeah. So, what you mean here is about the soil conservation, yeah. Especially you concerned about soil conservation, right?
Yes, conservation.
Okay.
Yes,
Soil conservation and
and sustainability.
as I understood that people also try to sell the soil material. Is it what you mean? Does it
Yeah,
what you mean?
the
Or-
soil itself, the land itself, the land itself.
Oh, you mean the land?
Yeah,
The land.
itself. Yeah,
Okay.
the land itself.
In the land use, yeah, in the land use
Yeah, the land
level. Okay. Yes. Yeah, it's. Yeah, it's a, once again, it's a big challenge. Yeah, it's a big
Yeah,
challenge,
big challenge,
Mr.
yeah.
Hadi. Yeah. So I think but this is the very. I mean, I would like to underline one point, just only one point. It is related to the regional planning map, or even something related to how the people or even the government can be strict, to the land use management. For example, which area that the people couldn't sell it anymore, and then should be protected, for example, and should be
Yes, it's
classified
correct.
into some categorization, right? So, I mean, this is the more important thing that the government should concern on. This also should focus on in this area. I mean, yeah, because if the regulation couldn't handle this kind of situation, means that the people can just do anything what they want. So that's why, yeah, the very last one that what I would like to say is about how the government, even all of the stakeholder, can manage in the better way and the agreement with the local people maybe I forget to say something in my presentation because it is related with the access, the access of the forest. This is still big homework for us, for all of us. This is also about one section on my dissertation, that how the people can manage the access to the forest, because the forest is still, I mean, the forest belongs to the government, but the local people already or have been working there for quite 100 years. You know what I mean? So this is about the access, basically. Yeah. So that's what I can say. So we need to re-strategize or revitalize about the regulations in terms of access and land use system management.
Yes. Thank you very much.
Yeah, I think it's also correlates with the Miss Puspitasari question about the deforestation and also the needs for housing, for example, and then the efforts to preserve the use of traditional plants. Yeah, Miss Astuti, maybe you can give further comments regarding this.
Okay, it is from Miss
Puspitasari.
Miss Puspitasari. Yeah. Okay, so I try to underline. Try to re-mention what I already said. Yeah. That's why it's a bit hard for me to say something, because this is our problem. I mean, our common problem. This is. What I can say, this is something in front of, in our yard. It's already in our yard, in front of us. So this is the real problem. So what I can suggest in this case is about to find the what is that, like another space here that we can also use it for making house, for example. So we can substitute it. And then, of course, we also can involve. We already have here University of Jember. We already have another university also, so we can collaborate. I mean, between the government, the regional, either Jember government, for example, and regional government, East Java in the level of regional government, and then all of the university here, and then the national park or even the stakeholder which is concerning or which is managing the forest area. So I think all of us can talk together. How could we solve this kind of problem? Because then local community they feel that they used to make housing by using the spaces from the forest. It's common, right, in their tradition. So yeah, the only way that we not the only way, I think. I mean, one of the best solution is just try to find out the substitute for the species, and then the other one, because some species also need another species. I mean, for example, like some herbs need another tree to live inside of the forest. Means that we can also domesticate this kind of species either in the buffer zone area or even in the rural.
Yeah, I think there is a connection problem with Miss Sri Astuti. Yes, I think.
But you can still hear my voice, Miss Afifah?
Yeah,
yes?
you can.
Yeah, okay.
So I can hear it.
Yeah, okay. And the last one is about what I said. Domestication. We also have a biosphere reserve, Blambangan, right? Correct me if I'm wrong, please. The name Blambangan Biosphere Reserve, as far as I know. And I also intend, maybe I can, after I finish my study, then I can help. Inshallah, I can help to develop, to develop and to develop or improve more, or even like how can we just upscale this management into the landscape area? Yeah, inshallah, I will do my best, to contribute my my thought, my everything to this kind of, this is my my dream also, that I can work in this landscape level, especially in Jember, because this is a long story. I'm the alumna also, or even my study from magister or even PhD, I also do the same thing there. I mean, the same area. So it was like long story, and like it has, I mean, I have a lot of memory about it. So yes, that's what I can say, and I can suggest. Thank you.
Yeah, thank you, Ms. Sri Astuti. I think it's also not only responsibility for the researcher, but also government, and also we don't forget it's also responsible for the community itself to conserve our world, and also for in sake of medicinal plants, I think. So if there is any participants who want to ask to our speaker this afternoon, please raise your hand. Okay, let me see.
Can I ask to Pak Amrun again one more question?
Yes, please.
Okay.
Please,
If nobody asking.
Sorry. Of course.
Okay. Okay. Pak Amrun, I work in physics, in biophysics, that maybe is sensor is in also in mind of my thing, but difficulty is for the calibration. I think is for yours is easy one for the calibration. The calibration of the to calibrate of your I mean your measurement, or your equipment, or your sensor. The process of calibration. Thank you.
Okay. First of all, using if we use to calibrate the scanner, flatbed scanner, we use what we call the color degradation block, or color degrading block supported by I don't know, I forgot, by Canon or which one, the other printer, Epson. There are color degradation to calibrate black to white. But if you mean that the calibration is a calibration curve to measure or to calculate something, every purpose has its own calibration substances. For example, if we want to determine total polyphenol content of coffee, which we know that chlorogenic acid is a major polyphenol found in coffee samples, whether green coffee, predominantly in green coffee or the roasted coffee. So we use chlorogenic acid as a standard. We characterize our sensor first before it is applied to coffee samples. So basically, characterization here is validation. So we made a calibration curve that measure linearity response between the chlorogenic acid and the color intensity of sensors. And then we also calculate the detection limit, or LOD. We also determine limit of quantification. We also test the selectivity. We also test many things in validation. So after we show that the sensor is okay in the characterization step, then we know that the sensor is ready to apply for a real sample determination. So when we measure the antioxidant capacity of curcuma genus, for example, maybe we can express the antioxidant capacity as a gallic acid equivalent, calculated as gallic acid equivalent. But it is not, I don't recommend, since the most polyphenol found in the curcuma genus, or let's say curcumin or temulawak, which is known as curcuma santoriza, all the curcuma rhizome or curcuma genus, the major polyphenol found is curcuminoids, or let's say the curcumin. So if we calculated the antioxidant capacity, we should calculate it as a curcumin equivalent, which is related or represented what compound, what major compound in that particular extract. I hope it answer your question, Bu Ari.
Yes, thank you very much. It's complicated, but it's complete. I think it's complete seminar to the conference. There is a method. There is the antioxidant. There is people. There is humanity, and there is community. It's very good. I think. Thank you very much for the committee and for the university and everybody, and then Sri Astuti Pak Amrut that here. It's very good. I think. Okay. Thank you very much.
Okay, thank you very much for your compliments, Bu Ari. And I'm still waiting one or maybe two that who want to ask by live by raising your hand. I don't think so. Maybe I can ask Ms. Sri Astutik for your closing statements, and also Mr. Amrun. Please first from Ms. Sri Astuti.
Okay, thank you so much, Ms. Afifah. Yeah, I think in this occasion, I would like to use this opportunity to say that basically in our life we need to marry between conservation and development, especially in terms of medicinal plants management. So we don't need to spirit both of them, because while we are by using both of concept, it could maintain, or it could sustain the medicinal plants for the long run. That's what I can say. And the very last one, all of the stakeholders of the medicinal plants resources should work together and hand in hand, and then especially we need to concern a lot for the community, the local community, because they are the key actor of the development process and development process, or even the conservation aspects. Thank you so much.
Okay, thank you. And so, Mr. Amrun, please give your close statement.
Okay, thank you, Dr. Aviva. I think since the paper-based sensor is developed or fabricated from paper, so it can be easily handled, and it is more safe to the environment, and it is also shoot with clean chemistry spirit. Whether we know that in the paper-based sensor, we only use a small amount of a regions and or bioregion, and also we also use a small amount or small volume of sample or standard solution. In this case, we already to reduce any chemicals that used, which is though it is more less what we call cemaran or contamination to the earth, to our beloved earth. Thank you, Dr. Afifa.
Okay, thank you very much for our both speaker in this afternoon. Very interesting topics. And I'd like to wrap up our plenary session this afternoon. So anything that may be a challenge, maybe it can trigger our future collaboration and our future research. Okay, thank you very much for all participants. So just giving back the time to the moderator.
Thank you so much to Apotheker Afifa Makhlourin, Master of Science, for guiding the last plenary session so well. Thank you once again.
The following agenda is awarding certificates to the speaker and moderator of the plenary session 4. The first certificate is for Ms. Sri Astutik, Master of Science, PhD candidates. Thank you very much for making the time today, ma'am. The second certificate is for Dr. Apotheker Muhammad Amrun Hidayat, Master of Pharmacy.
Thank you very much.
Thank you very much, Doctor.
And the third certificate is for Apotheker Afifa Makhlourin, Master of Science.
Thank you.
Thank you so much, ma'am.
Moving on to our last agenda, with honor, we would like to invite Dr. Apotheker 15 Aprila Fajrin, the Vice Dean 1 of the Faculty of Pharmacy, University of Jember, for the announcement of Best Oral Presenter.
And with honor, we would like to invite Dr. Apotheker Dian Agung Pangaribowo as the Vice Dean 3 of the Faculty of Pharmacy, University of Jember, for the announcement of Best Poster Presenter.
To Dr. Apotheker 15 Aprila Fajrin and Dr. Apotheker Dian Agung Pangaribowo, the stage is yours.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Finally, we arrive at the end of the event, our first international conference on medicinal plant ICMP 2022. Before closing this conference, we will announce the winners of the oral presenter and also the poster presenter. And we have 3 winners for each category. So let's start with announcing the winners in the oral presenter category. The committee is ready? Okay. The third winner belongs to Destria Indahsari with the title Physical Characteristic and Antioxidant Properties of Aquilaria microcarpa leaf extract gel after storage. Congratulations to Destri and Dasari. And we move to the second winner, belongs to Nuruli Vivi Anggraini, with the title Isolation Ligninolytic Bacteria from Rainbow Forest, Aijan Geoprak, Jonbo Bondowoso. Congratulations to Nuruli Vivi Anggraini. And the last but not at least, the first winner belongs to Mutia Hardiyuna, with the title Curcuma santoriza aqueous extract enhances pro-inflammatory cytokines, TNF-alpha and interleukin-6 gene expression in raw 264.7 macrophage cells. Congratulations once again to Mutia Hardiyuna, and of course congratulations for all the winners.
Now we move to the announcement of the winner in the poster presenter category. The third winner belongs to 15 Aprila Fajrin, with the title Antioxidant and Anti-Hyperalgesia Activity of Ethanol Extract and Fraction of Red Ginger in Early Painful Diabetic Neuropathy Mice. Congratulations. And then the second winner of the best poster presenter belongs to Yudi Purnomo, with the title Antioxidant Activity of Pulutan Urena lobata leaf fraction and Analysis of their Total Phenol Content. Congratulations for the winner. And now the first winner of the best poster presenter is Yuni Mumpuni, with the title Anthelmintic Activity of Methanol Extract of Jaha Leaves against Caenorhabditis elegans. To all of the winners, please contact the committee by email icmp@unech.ac.id. Once again, we congratulate all of the winners and all of the participants in the conference. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Once again, congratulations to the best oral and poster presenters for the first international conference on medical plants. What a wonderful creation. Your work hard is paid off.
Now, with honor, we would like to invite the committee chairman of ICMP, Apotheker Enda Puspitasari, Master of Science, to give a closing speech for the first ICMPs. To Apotheker Enda Puspitasari, Master of Science, the platform is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. On behalf of the organizing committee, I am delighted to deliver these closing remarks on the success of the first International Conference on Medicinal Plants, which will held hybrid. The committee successfully hosted this event as the plenary, parallel sessions, and all presentations were delivered with minimum technical issues. I believe that during these 2 days, we have all had insightful, interactive discussions and a great chance to share the outcomes of our research. Furthermore, I would like to thank our distinguished keynote speaker, plenary speakers, and the invited speakers who shared their valuable experiences. The working group on medicinal plants and traditional medicines, all the presenters, authors, reviewers, sponsors, and all parties who have contributed their best effort and assistance in organizing the event and ensuring the success of it. I also would like to congratulate the winners of the best oral and poster presentation. Although this is the end of the conference process is not over yet. There are still some steps that should be done, especially regarding your paper publication. The committee would like to remind those who will publish their article in our partner journals or our proceedings, full paper submission is awaited up to the 7th, which is 29th of October 2022. Otherwise, the articles will not be processed. As additional information, we would like to inform you that the proceedings required a minimum of 10 to 15 articles for the ISBN issuance. Thus, if the specified quota is not reached, the proceedings cannot be published. Regarding this matter, we will select the papers and check its suitability to your journal selection. However, we cannot guarantee that your paper will be published by the journals, because the final decision will be decided by the journals themselves. The waiting time will also be various from one journal to another. Therefore, we kindly ask for your patience. I do hope that the first international conference on medicinal plants is able to impact all of us to greatly foster and empower society through science and technology for sustainable innovation. Finally, I would like to thank all the committee members for organizing this conference and ensuring that it proceeded smoothly. I would also like to apologize to all the participants for any inconvenience that you might experience during the conference. To conclude, thank you for your great contribution and looking forward to seeing you at our next International Conference on Medicinal Plants. Thank you. Wassalamualaikum warahmatullahi wabarakatuh. Hereby, the first International Conference on Medicinal Plants is ended.
Recording stopped.